All right, let's go.
Number 14. The Black Walls. In 1991, 5 years after Chernobyl's reactor number 4 exploded, a robot sent into the ruins to survey the damage came back with something clinging to its surface that nobody expected: thick, black fungal growth. Inside a structure radioactive enough to kill an unprotected human in minutes, something wasn't just surviving, it was spreading. Researchers who later sampled the reactor walls and cooling water identified several distinct species, including Cladosporum sphaerospermum, Cryptococcus neoformans, and Wangiella dermatidides, all sharing one obvious trait: they were heavily melanized, meaning their cell walls were dense with the same dark pigment responsible for human skin color. Scientist Ekaterina Zhdanova, studying fungal samples from the accident site, documented something even stranger than simple survival. When exposed to beta and gamma radiation in controlled conditions, these fungi grew directionally toward the radiation source rather than away from it, a behavior she named radiotropism. That detail is what separates this from ordinary extremophile biology. Bacteria surviving in a hot spring or a salt flat are tolerating conditions that happen to exist there. An organism actively growing toward the most lethal radiation in the room is doing something that looks, on its surface, like the exact opposite of self-preservation, unless the radiation itself was somehow useful to it, which is precisely the question the rest of this list exists to answer. Number 13. The Radiation Eaters. In 2007, a team led by Ekaterina Dodacheva at the Albert Einstein Medical College in New York published a study in PLOS One that gave radiotropism an actual mechanism. Dodacheva's team exposed melanized fungi to gamma radiation and measured their growth against genetically identical non-melanized control samples kept in identical conditions but shielded from radiation. The irradiated melanized fungi grew significantly faster. The proposed process is called radiosynthesis, and the comparison researchers use deliberately echoes photosynthesis. Where plants use chlorophyll to convert light into chemical energy, these fungi appear to use melanin to convert ionizing radiation into a usable energy source. Dodacheva's team found that gamma radiation measurably altered the electronic structure of the fungal melanin, changing its oxidation state in a way that coupled to the cell's own energy production. A related 2011 follow-up examining Cryptococcus neoformans specifically found the irradiated melanin also changed its magnetic properties in the process, evidence of a genuine chemical transformation rather than simple heat absorption. None of this makes these fungi immune to radiation in any absolute sense. Extremely high doses still kill them, the same as anything else. What the research shows is a genuine metabolic pathway, this specific and this well documented, that appears to treat moderate ionizing radiation less like a threat to defend against and more like a resource to extract energy from. Number 12. The Toughest Bacterium. Long before Chernobyl, in 1956, American researcher Arthur Anderson and colleagues were trying to sterilize canned meat using gamma radiation when they discovered something had survived the process anyway: a pink spherical bacterium, later named Deinococcus radiodurans. Since then, it's earned a fairly literal nickname in the scientific literature: the toughest bacterium in the world. The numbers explain why. D. radiodurans can tolerate acute radiation doses up to roughly 5,000 gray with no loss of viability, a dose 3,000 times higher than what would kill a human and around 250 times higher than the threshold lethal to E. coli. At doses that shatter its DNA into hundreds of separate fragments, comparable to what a human genome would look like after a similar dose, D. radiodurans simply reassembles the pieces and keeps functioning. For decades, researchers assumed the answer had to be an unusually tough genome. Genomic sequencing published in 1999 found nothing unusual about the DNA itself. It breaks exactly as easily as any other bacteriums. What turned out to be exceptional wasn't the DNA, it was the repair system built around it. D-radiodurans runs a highly efficient recombination-based repair pathway using a protein called Rec-A to methodically stitch shattered chromosome fragments back into a coherent genome multiple times faster and more accurately than comparable systems in ordinary bacteria, turning what should be a fatal genome-shredding event into something closer to a routine maintenance problem the cell is simply built for. Number 11. The protein shield. For years, the DNA repair explanation for Deinococcus radiodurans seemed complete on its own. Then, researchers noticed something that didn't fit. Bacteria with comparably efficient DNA repair systems exposed to the same radiation doses still died, while Deinococcus radiodurans kept surviving. The genome wasn't the only thing that needed protecting. Follow-up research redirected attention towards the cell's proteins rather than its DNA. Radiation doesn't just fracture DNA directly, it also generates a flood of reactive oxygen species, unstable molecules that go on to damage proteins throughout the cell, including the very repair enzymes needed to fix the DNA in the first place. Deinococcus radiodurans, it turns out, maintains an unusually high internal ratio of manganese to iron ions, along with small manganese-based antioxidant complexes that neutralize reactive oxygen species before they can wreck the cell's protein machinery. Without that protection, later studies found, even a perfectly intact, undamaged DNA repair system would simply fail to function because the enzymes carrying out the repair would themselves be too damaged to work. The current scientific consensus builds from this line of research, reframes the entire question. Deinococcus radiodurans doesn't survive radiation primarily because its DNA is hard to damage. It survives because it protects the protein machinery responsible for cleaning up the damage afterward, meaning the real secret to surviving inside a radioactive environment may have less to do with defending the genetic blueprint and more to do with keeping the repair crew alive and functional long enough to use it. Number 10. The Vent Archaeon. Deinococcus radiodurans isn't actually the most radiation-tolerant organism known to science. It's simply the most famous one. That title belongs to Thermococcus gammatolerans, a single-celled archaeon first isolated in 1991 from a hydrothermal vent nearly 2,600 meters deep in the Guaymas Basin off the coast of Mexico. Living at temperatures above 80 degrees Celsius near vents naturally rich in radioactive elements and heavy metals, it can withstand acute gamma radiation doses of roughly 30,000 Gray, 6 times higher than Deinococcus radiodurans, and a dose that would kill a human more than 6,000 times over. A full genome and proteome analysis, published in Genome Biology in 2009 by a French research team led by Ivan Zivanovic, sequenced its entire genetic code specifically to locate the source of that resistance. The results were, in their own way, more interesting than a clean answer would have been. The genome contained no unusually large collection of known DNA repair genes, and no obvious structural trick setting it apart from closely related, far less radiation-tolerant archaea in the same genus. The study's own conclusion was that T gamma tolerance's extraordinary resistance likely comes from proteins that haven't been identified or characterized yet. That's a genuinely unusual place for this list to sit, this deep into researching the subject, the single most radiation-resistant organism confirmed on Earth, and the mechanism behind its defining trait remains, as of the most recent published research, still not fully understood. Number 9. The water bears. Tardigrades, microscopic invertebrates, sometimes called water bears, can survive radiation doses between roughly 4,000 and 5,000 Gray, comparable to Deinococcus radiodurans and around 1,000 times more than a mammal can withstand. For years, researchers assumed this was simply a side effect of a different survival trick entirely: tardigrades' well-documented ability to nearly eliminate all water from their bodies and enter a dormant state called anhydrobiosis, since dry tissue generally suffers less radiation damage than hydrated tissue. But later studies found some tardigrade species remain highly radiation tolerant even while fully hydrated and active, which meant desiccation alone couldn't be the whole story. In 2016, a Japanese research team sequencing the genome of the tardigrade species Rhamazottius variornatus identified a completely novel protein unlike anything found in any other organism, which they named DSUP for damage suppressor. When researchers inserted the gene for DSUP into ordinary human cells growing in a lab dish, those cells became measurably more resistant to radiation damage and hydrogen peroxide exposure than untransfected cells. Follow-up structural research has since shown DSUP works by binding directly to nucleosomes, the spool-like structures DNA wraps around inside a cell's nucleus, physically shielding the wrapped DNA from hydroxyl radicals, one of the most damaging byproducts radiation generates inside living tissue. It's one of the only radiation defense mechanisms on this entire list that's already been shown to function, transplanted whole inside human cells specifically. Number 8. The cooling pond catfish. Outside Chernobyl's reactor number 4 sits a cooling pond that supplied water to the plant, and it's home to something that draws visitors and documentary crews decades after the disaster: Wells catfish, some growing to sizes well beyond what the same species typically reaches elsewhere, cruising through water that once carried reactor cooling flow. Measurements taken from these fish have found radioactivity levels reported as high as 16 times normal background readings. The unsettling part isn't the radiation reading itself, it's that the fish, by every available measurement, appear largely unharmed by it. Wells catfish are naturally long-lived and can grow to enormous sizes under favorable conditions regardless of location, and much of their apparent size in the exclusion zone likely reflects the complete absence of fishing pressure for 40 decades rather than a radiation-driven growth effect specifically. What is genuinely notable is that a population living for generations in a pond with measurably elevated radioactivity hasn't collapsed and continues successfully reproducing. Sustained exposure that would represent a serious occupational hazard for a human, apparently absorbed by the fish as a survivable, if not entirely consequence-free, condition of daily life. Researchers studying the broader Chernobyl aquatic ecosystem have generally found similar patterns across other fish species in the zone's contaminated waterways: measurable bioaccumulation of radioactive isotopes in tissue without the outright ecosystem collapse that early post-disaster predictions widely assumed would occur. Number 7. The thriving count. For years after 1986, the working assumption among most ecologists was straightforward. The Chernobyl exclusion zone should be a biological dead zone, its radiation levels too dangerous to support meaningful wildlife populations for generations. In 2015, a long-term census published in Current Biology by an international research team, including biologist Jim Beasley, delivered results that directly contradicted that assumption. Aerial and camera trap survey data showed populations of moose, roe deer, red deer, and wild boar within the zone at levels statistically similar to 4 uncontaminated nature reserves in the surrounding region. Wolf populations were found to be roughly 7 times higher than in those same comparison reserves. The explanation the researchers settled on wasn't that radiation was somehow beneficial to these animals. It was that the near-total removal of human presence, no hunting, no farming, no logging, no highway traffic, outweighed the biological cost of chronic low-level radiation exposure by a wide margin. As team coordinator Jim Smith put it directly, the results don't mean radiation is good for wildlife, only that the accumulated damage humans do to an ecosystem through ordinary habitation turned out to be considerably worse than living inside one of the most radioactive landscapes on Earth. A follow-up 2016 camera trap study using remote scent stations across the zone reinforced the same conclusion, finding animal distribution patterns that showed no measurable correlation with local radiation levels at all. Number 6. The counter-evidence. Not every research group studying Chernobyl's wildlife agrees with the thriving population narrative, and the disagreement is genuinely unresolved rather than settled in either direction. Evolutionary biologists Anders Mahler and Timothy Musso have spent over 2 decades conducting a separate, extensive body of field research across the exclusion zone, publishing dozens of peer-reviewed studies that reach a considerably darker conclusion. In one 2007 survey, they counted 66% fewer birds and 50% fewer bird species in highly radioactive locations compared to background-level sites, alongside significantly reduced numbers of insects, spiders, and small mammals, including hares and foxes. Their research goes further than simple population counts. A 2011 study led by Mahler found bird species living in more radioactive areas of the zone had measurably smaller brains, a roughly 5% reduction on average tracking radiation level, even after controlling for overall body size, which the team linked to chronic oxidative stress interfering with normal neurological development. In a related finding, Musso's team reported that up to 40% of male birds sampled in the most heavily contaminated areas produced deformed or non-viable sperm, rendering them functionally sterile. Researchers on both sides of this disagreement acknowledge the exclusion zone contains wildly varying radiation levels across different microlocations, which makes broad conclusions genuinely difficult, and a 2022 review in Knowable magazine described the dispute as still actively unresolved among scientists studying the same landscape. What both research groups agree on, despite reaching different conclusions about overall wildlife health, is that low-dose radiation is measurably altering biology in the zone in ways that are still being mapped, not simply ignored by the organisms living there. Number 5. The darker frogs. Not every adaptation inside the exclusion zone is invisible at the molecular level or contested between research teams. Eastern tree frogs living within Chernobyl's most contaminated areas have been found across multiple field studies to be measurably darker in color than frogs of the same species living in low radiation areas nearby, and the pattern tracks radiation intensity directly, with frogs from the most contaminated sites showing the darkest coloration. The proposed mechanism circles back to the same pigment already covered twice on this list, melanin. In frogs, as in the radiotrophic fungi from earlier entries, melanin appears to offer a genuine radioprotective benefit, potentially by neutralizing free radicals generated by ionizing radiation before they can damage cellular structures, in addition to its more familiar role in coloration and camouflage. Researchers studying the frog populations have proposed this represents a case of rapid evolutionary selection, playing out over a comparatively small number of generations since 1986, favoring individuals whose naturally darker pigmentation happen to confer a survival advantage in a chronically irradiated environment. It's a genuinely rare opportunity in evolutionary biology, a population under a specific, identifiable, and measurable selective pressure, with a visible physical trait tracking that pressure in a documented, reproducible pattern, all unfolding within a human research timescale, rather than across the geological timescales evolutionary change usually requires study of. Number 4. The wolves' genes. The most recent and least settled entry on this list comes from Chernobyl's wolves specifically. In 2024, researchers led by biologist Kara Love and evolutionary biologist Shane Campbell-Stoughton entered the exclusion zone and collected blood samples from wolves living there, comparing them against wolf populations in nearby, lower radiation areas of Belarus, and against a baseline population from Yellowstone National Park in the United States, where background radiation sits at ordinary Earth normal levels. The comparison found 3,180 genes behaving differently in the Chernobyl wolves compared to both other populations, a striking number, though the research remains in an early stage, first presented at a scientific conference in 2024, with a formal paper still in preparation as of this recording. The team has been cautious about overstating conclusions this early, but the specific genes involved include several associated in other species with cancer resistance and immune system regulation, precisely the biological systems radiation exposure would be expected to place the heaviest selective pressure over successive wolf generations living inside the zone. Campbell-Stoughton has been explicit about where this research is heading next, active collaboration with cancer biologists, specifically to determine whether any of the genetic differences found in Chernobyl's wolves point toward mechanisms that could eventually translate into new approaches for human cancer treatment. Number 3. The space experiment. The radiotropic fungi first identified growing on Chernobyl's reactor walls have since left the planet. Cladosporium spherospermum has been sent to the International Space Station specifically to test whether its radiation-absorbing properties could offer meaningful protection to astronauts who face significantly higher background radiation exposure in orbit than anything experienced on Earth's surface without the planet's atmosphere and magnetic field providing their usual shielding. The proposed application isn't turning astronauts into fungal colonies, it's structural. Researchers have proposed a relatively thin layer of the fungus, grown as a living, self-replenishing coating, could function as a supplemental radiation shield on spacecraft or habitat walls, potentially self-repairing and continuing to grow as long as it's supplied with basic nutrients, unlike inert shielding material that simply degrades over time with no ability to renew itself. Early results from the ISS experiments found measurable radiation attenuation through a fungal layer, though researchers are still working out exactly how thick a functional shield would need to be for meaningful astronaut protection on a longer mission, such as a multi-month transit to Mars. It's a fairly direct, practical answer to a question this entire list keeps circling. If an organism evolved by accident inside the most radioactive man-made environment on Earth, it may already be pre-adopted for one of the most radiation-heavy environments humans are trying to reach next. Number 2. The human question. None of the mechanisms covered on this list, radiosynthesis, Deinococcus radiodurans manganese protective repair system, whatever unidentified protein defends thermococcus gamma tolerance, DSUP or adaptive melanism, currently translate into humans being able to safely tolerate meaningfully higher radiation doses than we already can. Human radiation safety standards are built around the linear no-threshold model, which assumes cancer risk from ionizing radiation increases proportionally with dose, all the way down to 0, with no dose considered entirely risk-free. That model itself remains a genuine point of active scientific debate rather than settled consensus. A competing framework called radiation hormesis proposes that very low doses of radiation might trigger beneficial cellular stress responses, upregulating the same kind of DNA repair and antioxidant machinery this list has repeatedly described in fungi, bacteria, and tardigrades, potentially making extremely low-dose exposure less harmful, or in some disputed studies, marginally beneficial compared to what a strict linear model would predict. The hormesis hypothesis has never achieved the kind of broad replicated consensus needed to change official radiation safety guidelines, and most major regulatory bodies continue to apply the more cautious linear no-threshold standard specifically because getting this wrong in the direction of underestimating risk carries much higher stakes than being overly cautious. What's genuinely being pursued, rather than the abandoned idea of raising human radiation tolerance directly, is transplantation, taking specific, well-understood mechanisms, DSUP being the most advanced example, already tested inside human cells with a measurable protective effect, and asking whether they could be safely introduced as a targeted temporary defense for specific high-exposure situations, like long-duration spaceflight or radiation therapy. Number 1. What survival actually means. Line up everything on this list, and the picture that emerges is more layered than a simple story of life triumphing over radiation. Fungi surviving inside Chernobyl's reactor repurposed melanin, a pigment nearly as old as complex life itself, toward a genuinely new metabolic function. Deinococcus radiodurans evolved an unusually good DNA repair crew, plus the antioxidant protection needed to keep that crew functioning under fire, and thermococcus gamma tolerance outsurvives it by a factor of 6 using a mechanism nobody has fully identified yet. Tardigrades evolved a protein that physically shields their DNA from damage before it happens. Chernobyl's frogs turned existing pigment variation into a measurable survival advantage within a few dozen generations. Its wolves may be quietly rewriting their own cancer resistance genes in real time. But the same landscape that produced all of that also produced Muller and Musso's 2 decades of evidence for smaller-brained birds, silenced insect populations, and sterile sperm, a body of research that hasn't been resolved against the more optimistic wildlife census data, only left standing alongside it, unreconciled. Radiation didn't select for a single outcome here. It applied pressure indiscriminately to every organism it touched, and different lineages, sometimes within the very same exclusion zone, responded by adopting, by declining, or in a genuinely small number of documented cases, by finding a way to use the exposure itself. None of this describes life becoming immune to radiation. It describes existing biological tools and the limits of those tools being tested against an environment more hostile than nearly anything most of these lineages had ever previously encountered, with the results, 40 decades in, still being counted. 14 organisms, and no single story ties them all together cleanly. A fungus that treats radiation as food. A bacterium whose real secret turned out to be protecting its repair crew rather than its genome. An archaeon that outlasts all of them by a method nobody has identified. A tardigrade protein already working inside human cells. Catfish and frogs and wolves living inside the same exclusion zone. And 2 research teams who, after decades of separate fieldwork in that same zone, still don't fully agree on whether the wildlife there is thriving or quietly suffering. What connects every entry on this list isn't a triumphant answer. It's proof that when biology is pushed this hard, it doesn't respond in only one direction. Subscribe to Cosmic Hives. See you in the next one.